I know that I have not been posting videos every day, even though the Summer Everyday Science Challenge has been going on, and I am so sorry about that. But I have had COVID for a few weeks now, and I am still recovering. I just cannot kick this virus. And I thought it would be a good opportunity to talk about how these at-home COVID and flu tests work, because the technology is the same technology we use in the research labs that I work in to detect different pathogens. And so I think it's really cool that you get to do this at home and be a microbiologist from your own kitchen. Now, these tests work by first collecting your sample. We get ours with these nasal swabs. You swish them around in your nose, and the swabs have little fibers that will pick up viruses and bacteria and sometimes boogers and that kind of thing. And then you put it in a special solution called a lysis buffer. And this liquid has certain chemicals in it that are going to break apart viruses and release the proteins that are in those viruses. And those proteins will be floating freely in there. And those proteins are what we use to identify specific viruses. Every virus has its own specific signature of proteins, and your body knows this as well. That is how your immune system works. You have antigens in your body that will detect these proteins. It's sort of like your antigen is a lock and the protein is a key, and when they bind together, they create a reaction. And in your body, that reaction causes an immune response where your body will start attacking that virus. In this cartridge that we have here, it will actually cause a color change. So this cartridge has very special paper on it, and when you drop the liquid on it, it moves through using capillary action. So it's like a paper towel sucking up that liquid. And as that liquid is moving through this special paper, the paper has antigens for each of these viruses on it, for flu A, for flu B, for COVID. And if you have any of those proteins in your sample that you are putting on here, it will bind to those antigens that are stuck on the paper and cause a color change. And you can identify which viruses you might be infected with. And there is a control line on here that will always change color every time you add this special lysis buffer on there, just so that you know the test is working properly, even if you don't see any of the virus lines. So I'm hoping to feel better soon and start posting everyday science videos again, and I really hope that until next time, you keep discovering.